In today's video, I'm going to be continuing on the topic of enzymes, but today we're going to be looking at enzyme-controlled reactions. This video is very important in learning some of the vital skills that will help you answer both experimental and application questions that may be asked in your exam. Now, some of the experiments I describe today you may be completing in class as part of your practical assessment for biology. However, they can also come up as either experiment-based questions in your exams or application questions. And application questions are essentially questions where they build on the prior knowledge you should already have from your syllabus, and they will ask you a question that expects you to use your knowledge and apply it in a slightly unfamiliar context. So let's have a recap on some of the basics we learned about enzymes in the last video. If you haven't watched that video, I have linked it in the description, and I would strongly suggest watching that first and then continuing on and watching this video. So we can see here we've got a diagram that shows all the stages of enzyme and substrate bonding and the breakdown of substrate into products. So enzymes contain an active site which binds a specific substrate. Normally enzymes only have one complementary substrate. Sometimes they can have multiple, but for the most part, A-level biology is only going to be asking you about enzymes in the context of it having one specific substrate. So we can see from our diagram that our substrate actually looks like it's going to fit into the enzyme's active site by the lock and key mechanism, which is a theory that suggests that substrates have the exact shape to be able to fit into their enzyme's active site. However, we now know that there's a slightly updated model that we use, which is called the induced fit model, which is where substrates will have a similar shape to their enzyme's active site, but will actually induce a conformational change in the active site so that the substrate is able to be held much more tightly within the enzyme's active site. Now, in this particular diagram, we can see that we've got one substrate, and that is being broken down into two products. However, that can happen, and substrate breakdown will happen through pressure that is being put on the bonds. However, we can have two or more substrates coming in and slotting into the enzyme's active site, and this will form one product, and that is because the active site is able to hold these two substrates in place and reduce the repulsion forces between them, allowing them to bond much more easily and form a product. So let's now go on and talk about an experiment we can use that will measure the rate of product synthesis in an enzyme-controlled reaction. So here we can see I've got an equation that shows 2H2O2, which is the formula for hydrogen peroxide. So H2O2 is hydrogen peroxide. And this is broken down into two molecules of water and one molecule of oxygen gas. And this reaction is catalyzed by an enzyme that is creatively named catalase. So we can see here we've got our experimental setup. And what we can see on the left-hand side is that we've got a flask, and it is in a closed system. There's a bung on top of the flask, there's a small hole so that we can put a thermometer in, and there is a hole that allows a gas delivery tube to come out of the flask. But other than that, nothing can escape. So really, the only place any gas formed will go is down the delivery tube. And we can see in the bottom that we have a solution of catalase, and we also have a small weighing bottle that will contain hydrogen peroxide. Now, this flask is held in a water bath, which will keep it at a constant temperature. And what will happen is that the flask is shaken slightly, which will cause the weighing bottle to tip over, and the hydrogen peroxide will mix with the catalase solution and will be broken down into water and oxygen. Now, oxygen being a gas will expand to fill its environment, so it will go down this delivery tube. And if we look to the other side of the delivery tube, we can see that we've got a vessel containing water, we've got an inverted measuring cylinder which is also containing water. And what will happen is, as oxygen is produced, it will travel down the delivery tube and it will displace the water that is found in the measuring cylinder. So what will happen in the reaction is that the weighing bottle will be knocked over and the hydrogen peroxide will interact with the catalase, and we will measure the production of oxygen. Normally it's over 1 minute, it might be slightly longer depending on the experiment that you're doing. And then what we can do is we can look at the average rate of product synthesis. So what we will do is look at the volume of oxygen produced. So let's just say in a minute, for very easy maths, we've got 6 centimetres cubed of oxygen that's been produced, and we divide this by the time taken. So in this example, 60 seconds, which will mean that we get 0.1 centimetres cubed of oxygen forming per second. So we can see we've got on the bottom of the screen there, we've got our units of measure. So centimetres cubed, seconds minus 1. If you see something and the index is minus something, that minus essentially just means per. So S minus 1 means per second. So we can do this experiment and set our water baths to different temperatures, and this will allow us to find the optimum temperature that catalase works at. So you may do it at 10, 20, 30, 40, 50 and 60 degrees, and you might get an increasing rate of oxygen forming all the way from 10 to 40 degrees, but at 50 degrees perhaps the reaction begins to slow down, and at 60 degrees it will stop, and this could be because at that temperature catalase is becoming denatured due to the high heat. So each time we repeat this experiment, we need to make sure that we are adding the same volume of hydrogen peroxide and the same volume of catalase, and this just means that we're only changing one independent variable. And this experiment should be repeated 3 times at each temperature so we can work out an average. Let's move on and look at a different experiment now, looking at the breakdown of substrates. So here on the left-hand side, you can see my badly drawn diagram. In our test tube, we have got a solution of starch and amylase. Now, if we remember back from our videos on carbohydrates, amylase is an enzyme that breaks down starch into maltose. And we can also see that we have a spotting tile set up. It's got all these different coloured circles. I will explain why they're different colours in a minute. But in our spotting tile, in each well, we put a few drops of iodine dissolved in potassium iodide solution. And as we know, we've met this agent before. So iodine dissolved in potassium iodide changes colour in the presence of starch. So when there's no starch present, it's this orangey brown colour, and when starch is present, it changes to this blue-black colour. So what we would do to do this experiment was that we would put a known concentration of starch and amylase into our test tube. Then we would pipette this solution into one well of the tile at regular intervals. So you could do it every 10 seconds. So in our first well, we'd put it in straight away as soon as they've been mixed, and then the next one at 10 seconds, then at 20 seconds, 30 seconds, etc. And as we just said, if starch is present, the iodine in potassium iodide will turn this blue-black colour as soon as we pipette the solution from our test tube in. However, when starch has completely broken down, obviously it will no longer turn blue-black, it will just remain this orangey-brown colour, and that means that the amylase has completely broken down all our starch in solution. So this test can show us how fast amylase is working by recording the time taken for the solution to no longer turn blue-black. And once again, we would repeat this 3 times to get an average of how fast it's working. And then we can look at changing some of the parameters of the experiment. So we can look at changing some of the variables. So in this experiment, we could increase the concentration of either our enzyme, so amylase, or our substrate, starch. We can look at it at different temperatures by keeping our test tube in a water bath and then pipetting out our solution, or we can look at changing the pH, so adding different types of buffers into our test tube as well. Now, if you were to be asked a question on this and it simply said, describe an experiment that measures the breakdown of a substrate, you would just essentially describe this experiment or perhaps another one you've learned about and just go through the steps. And at the end, you would say, the experiment needs to be repeated at 3 times using the same volume of starch and the same volume of amylase, just to make sure that you're not changing any independent variables. However, if it asks in a specific context, so maybe you get a question that says, describe an experiment that measures the breakdown of a substrate over a range of pH values, then obviously you'd adapt your question at the end, or your answer to the question at the end, and say something along the lines of, this experiment should be repeated 3 times every pH and different buffer solutions added to allow the pH of the starch and amylase mixture to change. Moving on to our third skill that we need to know about in today's video, we're going to be looking at a graph of an enzyme-controlled reaction and interpreting it. So we can see in the middle of the screen, I've got a graph that is showing the time of the reaction on the x-axis and the volume of the product that's being produced from our reaction on the y-axis. Now I've got 3 different lines here in 3 different colours. Our blue line shows the reaction occurring at 25 degrees, the red line the reaction at 37 degrees, and the green line showing the reaction occurring at 65 degrees. So what we do when we're interpreting a graph is that we would first measure the initial rate of reaction, which is this section of the graph that I've just highlighted with the box. And the initial rate of reaction is the rate of reaction that occurs at the time point 0. And we will look at how to do this in just a second on my next slide. And then what we would do after we've measured our initial rate of reaction is we will analyse what the graph is continuing to show us. So we have a look at each line. First of all, on our blue line, we can see that our rate of reaction is constant and not all the substrate is used up. And we can interpret this because a lot of our reactions that we learn about are happening biologically in humans, and our biological temperature, our internal temperature, is 37 degrees or roundabout. And so that is our optimum temperature that a lot of enzymes are designed to work at. So 25 degrees is colder than most enzymes would like. So both the substrates and the enzymes have less kinetic energy, and therefore they're less likely to collide because there's less movement. So we're less likely to get enzyme-substrate complexes forming. So eventually, if we extended our x-axis, eventually this line would plateau, because if we're not replacing our substrate, eventually it will get used up and then obviously no more reaction is occurring. But because this is over a limited time period, essentially we have a constant rate of reaction. Now at 37 degrees, we looked at what happens in our last video, but essentially our rate of reaction increases and then the graph will plateau because either all the substrate is used up or all the enzyme active sites become full. So the rate of reaction plateaus. Now at 65 degrees, we can see our graph is going to plateau much earlier. So obviously 65 degrees, the enzymes and the substrate have a lot of kinetic energy, they're vibrating a lot more, there's a lot more movement, so they're more likely initially to collide and form an enzyme substrate complex. However, because of the increased vibrations, the enzymes, the vibrations will cause the bonds in the active site to break. And this will mean that the active sites and essentially the enzymes denature, they're no longer complementary to the substrate, and we won't get enzyme substrate complexes forming, so the reaction will stop, which is why it plateaus so much earlier than at 37 degrees. So let's look now at how we can use tangents to calculate our initial rate of reaction. So we can see I've got a similar graph that's just come up on screen, looking at the volume of product that we produce over time. And what we do is, the first thing we do is extend the curve. Now normally what you would do is you would just extend the curve using a pen and a ruler underneath the axes. However, I can't do this on a computer, so using my limited technological skills, I've just overlaid another another curve over the top. Now we can see that this is not the perfect fit, it's not the perfect overlay, but for this example it will do. So you can see I've extended my line, and this is really important for our next step, where we draw a tangent at the point t equals 0, and it needs to be equidistant to the curve. So what this means is that it is flat against the curve and that our tangent line is coming away from either side of the curve on the same rate on both sides. It's quite difficult to explain without showing it on paper. Now obviously I'm trying to explain this off a PowerPoint slide. So it's much more difficult to explain, but I will try and see if I can link in the description box another video that maybe explains this better than I can, and it should be a skill that you learn pretty early on in class, so hopefully this explanation is just a refresher. So essentially equidistant, it touches the curve and extends away equally on either side of the point where it touches, and essentially this is just preventing it not being so steep on one side and perhaps crossing our curve on the other side. And what we do after that, once we've got our tangent drawn, is that we pick a point on the tangent and we're going to draw a horizontal and a vertical line that cross over the x and y axes. So this, if you've drawn your tangent correctly, you can pick any point on your tangent and it will give you the same result. So I've chosen, just to make the maths easy and to make sure I'm getting correct results, I have picked it going through 30 seconds on my x-axis and at 40 centimeters cubed on my y-axis. Now obviously I could pick a different point, it could be going through at 20 centimeters cubed on the y-axis and 15 seconds on the x-axis, but like I said, if your tangent is drawn correctly, you will always get the same result as long as you're interpreting correctly. Now I like to pick it going through the numbers on the axis, just because then there's no confusion. If it goes in between some of the numbers, you could be puzzling over, oh, is this 26 seconds or 27 seconds? So this just makes it much easier and much more accurate. And then what we do is we divide our y value by our x value. So I will be dividing 40 centimeters cubed by 30 seconds. So 40 divided by 30 is 1.33, and our units will be centimeters cubed per second. So from this graph, we can determine that our initial rate of reaction is forming 1.33 centimeters cubed of gas per second. So let's recap what we've just covered. We know that product synthesis can be measured using catalase to break down hydrogen peroxide, H2O2. Oxygen gas produced in a closed system will displace the water volume. We've also learnt that the breakdown of substrates can be measured by pipetting a mixture of starch and amylase into iodine dissolved in potassium iodide at regular intervals, and the rate of starch breakdown can be measured when the pipetted mixture turns the iodine dissolved in potassium iodide a brown-orange colour instead of the blue-black. We know that enzymes at an optimum temperature will have an increasing rate of reaction, but this will eventually plateau on a graph as the active sites become full or the substrate is used up. We know that above optimum temperatures, enzymes will denature and reactions will cease to continue. We've learnt that tangents can be used to calculate the initial rate of reaction, and that tangents should be drawn at the time point t equals 0 and be equidistant on each side of the curve. We've learnt that the initial rate of reaction is calculated by dividing the y-axis value by the x-axis value, and the example I've given is y value in centimeters cubed divided by x value in seconds will give z, so that's our result, z centimeters cubed per second. So now that we've learnt this information, it's important to go back, make sure you understand what's being explained completely, and then do the following test questions. So number 1, describe an experiment you could use to measure the rate of product synthesis. Describe an experiment that measures the breakdown of substrates. Try and do these questions without referring back to your notes and seeing that you've fully understood how these experiments take place. And number 3, describe the data shown in the graph and explain the trend of each line. Another important question that I've not included on this test yourself slide would be to go back through past papers and try find questions where they're asking you to draw tangents. I haven't given this because obviously this is taking place on a computer, and so obviously you can't really do a full measure, but perhaps printing out a page from a past exam paper where they've got a small bit of graph paper and you can work out how to measure a tangent. And then obviously once you've completed those questions, make sure you're going back, looking at the mark scheme and seeing that you've got it correct. And if you have, excellent. If you haven't, the best thing to do is to take it to your teacher or a member of staff or a friend and just work through the question to make sure you fully understand, because drawing tangents is quite a difficult skill. It's something I really struggled with when I did my A-levels, but it's a really important one to know. It almost always comes up on papers. So yeah, just making sure that you understand the skill. That's all for today. Thank you for watching.